Welcome to this session of Meeting Minds Global 2021, Oxford's COVID-19 vaccine and science making history. I'm Sally Benner in the Oxford North America office in New York City. This live broadcast is being recorded and will be available to you afterward. The session will last an hour, 45 minutes for conversation and 15 minutes for Q&A. You'll have a lot of questions and we'll try to get to all of them. Enter your questions in the Q&A box beside the video image. We encourage you to include your name and Oxford affiliation with your question. Unforeseen circumstances prevent author Walter Isaacson from conducting today's interview as originally planned. Mr. Isaacson is a Pembroke alumnus here in the United States, and we hope to feature him in another presentation in the future. Instead, William James, professor of virology, will lead the conversation with Professor Gilbert. From these two close colleagues, you will hear a story about Oxford's COVID-19 vaccine that you haven't heard before. You will learn about science that's making history. First, allow me to introduce a wonderful friend of Oxford Medical Sciences. Jonathan Turner, a British native, is a graduate of Magdalen College and is chairman of the college's investment committee. He is a longtime tech investor and advisor based in the Bay Area of California. Jonathan is a co-founder and former co-president of Catalyst Partners and has previously advised the UK government on technology and finance. In early 2020, Jonathan led efforts from key supporters to fund development and trials of Oxford's COVID-19 vaccine. Though it may sound cliche, the researchers couldn't have moved as swiftly and as boldly without such early philanthropic support. It's with great pleasure that I welcome Magdalen alumnus Jonathan Turner to this session of Meeting Minds, featuring medical sciences at the University of Oxford. Jonathan?
Thank you, Sally, and welcome to everybody calling from around the world. I first met Sarah Gilbert over Zoom, of course, in the dark days of March 2020, as she urgently needed funds to support her promising vaccine candidate. It struck me as a venture capital problem, how to provide funds before government and the commercial sector fully came to the table for a promising project with huge potential. We were fortunate to find alumni and some non-alumni who were willing to support the project. Today, Sarah's vaccine has been given to more than 32 million people in the United Kingdom and hundreds of millions around the world. You'll enjoy hearing the story. But first, some background on Sarah and Professor James. Sarah Gilbert holds the Saïd Professorship of Vaccinology and is one of the leaders of Oxford's COVID-19 vaccine development program. She joined the Nuffield Department of Medicine at Oxford in 1994 and became part of the Jenner Institute within NDM when it was founded in 2005. Her chief research interest is the development of viral vectored vaccines that work by inducing strong and protective T and B cell responses. Professor Gilbert leads the Jenner Institute program in influenza vaccine development and now also works on vaccines for many different emerging pathogens, including Nipah, MERS, Lassa, and Crimean Congo hemorrhagic fever. Using the viral vector platform she and her colleagues at Oxford have developed over many years, Professor Gilbert has been able to take new vaccines from design to clinical development, with a particular interest in the rapid transfer of vaccines into GMP, Global Good Manufacturing Practice, and first in human trials. 100 days after the genomic sequence of the COVID-19 coronavirus was released, Oxford's COVID-19 vaccine entered phase 1 clinical trials in the UK. This succeeded in record time due to her lab's many years of preparation and is a historic achievement by Oxford's medical scientists. William James is a professor of virology in the Sir William Dunn School of Pathology in the Medical Sciences Division at Oxford. His research ranges from stem cells to neurodegenerative disorders to HIV. He focuses specifically on tissue macrophages and how pathogens such as HIV circumnavigate these defense mechanisms. At the onset of the COVID-19 emergency, Professor James was integral to Oxford's SARS-CoV-2 virus facility, a pan-university core facility to test treatments to combat the coronavirus. Professor James is a tutor in medical sciences and formerly served as Pro-Vice Chancellor for Planning and Resources at Oxford. This unique Oxford conversation will explore science making history, how vaccines are developed, and where Professor Gilbert's research at Oxford is taking her next. It's my honor to introduce Professor Sarah Gilbert and Professor William James to the global Oxford alumni community. Saïd.
Thank you very much, Jonathan. That's an incredibly helpful introduction and very, very flattering. If I may turn to Sarah, thank you so much for making time available today from your incredibly busy schedule to talk to us. We're very grateful. I think many people in Oxford and elsewhere were surprised, actually delighted, but surprised that Oxford was so well prepared to meet the need for a vaccine for SARS-CoV-2. But in fact, you and your colleagues, as Jonathan was alluding to a little earlier, had been working on developing a vaccine platform that could tackle really tough problems like HIV and malaria and TB for many years. And I think you've been recently evaluating vaccine candidates to emerging virus infections such as Ebola and MERS-CoV. Could you tell us a little bit more about this journey and why it prepared your team so well for the emergence of COVID-19?
Certainly, I'd be happy to do that. And good evening, everybody. Good afternoon or good morning, wherever you are. And thank you very much also to Jonathan for his very kind introductions. And the role of philanthropic funding really was very important about this time last year. And maybe we'll just touch on that a bit later on, where that came in with we had some really unexpected funding needs as the program developed. And so we had some non-conventional asks for funding, which fortunately were met, and that was a really big help. So as you said, I've been working at Oxford for quite a long time now, came here in 1994, initially to work on the genetics of host parasite interactions in malaria, not vaccines at all, it was genetics. I've been working in a biotech company before then. I've been expressing human blood proteins in yeast, making proteins that potentially had therapeutic use. It was a biotech company that when I first joined was really expanding. It was an exciting place to work. Things were getting bigger and better and lots of new projects. But then the parent company decided to sell it. And so through no fault of anything that was happening in the biotech company itself, budget started to be cut, work started to become less interesting, good collaborations had to be ended. And I decided I'd have another look at academia, and because I was really interested in the science and wanting to be able to apply science for benefit, and thought I'd have another go at working it as an academic and see where that took me. And I haven't left Oxford since, really, apart from a lot of travel to scientific conferences. But the work on the genetics of how the genetics of the host and the genetics of the malaria parasite interact all comes down to actually immunology. It's an immune response that is responsible for how the host recognizes the parasite and what the parasite can do to evade that. And that gave us some clues about a new way that we could actually start to work on a vaccine against malaria. Okay. So that happened quite quickly after I'd moved to Oxford. And because I'd got experience in working with recombinant DNA and sort of making different things, I got involved in the vaccine development work. And at that time, DNA vaccines were quite new and a big thing, but we were also looking at viral vectors. And I started working, not with adenovirus initially, but with some other viral vectors, working out how we could make vaccines. And we were particularly interested for malaria in making vaccines that gave strong T-cell responses.
This is a very general audience, but could you try and explain to the audience in simple terms why the T-cell responses might be particularly important? not just for all pathogens, but for things like malaria in particular?
So for malaria, the stage of the life cycle that we were looking at was when the parasite gets inside the liver. So what happens if you get bitten by a mosquito that's infected with malaria and it injects parasites into the bloodstream as it takes a blood meal? Those parasites very quickly find their way into the liver and get inside a liver cell, and they make that their home for about a week, and they make lots of copies of themselves. And after that stage, they come out in very much larger numbers than originally went into the body, and start, actually, that's when the disease onset really starts. That's when you start to get the fevers. While they're hidden away in the malaria cells, in the liver cells, antibodies can't reach them. They're tucked away out of the blood circulation. So if you had antibodies, once they're inside a cell, it can't do anything. But the other part of the immune system that we don't hear so much about is the T cell response. And T cells are a bit like antibodies in that they have a specific target. They will recognize a cell that's infected with a malaria parasite in this case, and they'll destroy it. So when they
it's like a critical time in the malaria parasite's life cycle, really early on when it's establishing itself, you could target the cells it's infecting rather than the malaria parasite itself. Yeah. Okay, very nice.
That's a particular part of the malaria life cycle where if you can get in early and stop those infected liver cells from producing the hundreds of parasites that are inside there, germinating, if you like, then you can stop the disease in its tracks. But antibodies won't work at that stage. It has to be a T cell response. Most vaccines that induce T cells will induce antibodies as well, but there are some like the inactivated flu vaccine that are good for antibodies but not for T cells.
Okay, so what's the characteristics of the sort of vaccines that do the T cell part better than inactivated vaccines?
Those vaccines are ones that actually enable the body to make one of the proteins from the parasite, or the virus, as we'll come on to later, inside a cell as if there was an infection. So if you put recombinant protein or just pure protein into the body, you won't get a good T cell response. If you use what DNA vaccines, RNA vaccines, and viral vectors all have in common is that when you use them as a vaccine, they're carrying the gene to make a protein from the pathogen, the instructions to make that protein inside the body. And the vaccine goes inside a cell, that protein is produced, and then the immune system realizes there's something new here. This is something that is foreign, it shouldn't be here. And the immune system kicks in and starts to make a response against that protein because it looks like an infection. So all of these different technologies are fooling the immune system into thinking there's an infection. And there's this new protein. So when it's the coronavirus, which is the spike protein we're using for malaria parasite, it's one of the proteins that malaria normally produces. The body recognizes this is wrong, this protein shouldn't be here, and it kickstarts an immune response. And that results in the formation of immune memory. So the second time you get exposed to that same protein, which might be from the parasite itself or the virus itself, the immune system responds much more rapidly because it's seen it before. And that's what we're trying to do with vaccines. We're trying to form that immune memory against specific parts of a pathogen so that when you meet the pathogen, the immune system is ready to go. And it takes off really quickly with the right kind of immune response to protect you against the disease.
Oh, that's really helpful. And you mentioned you had a sort of a journey through different ways of priming such T cell responses, starting with, I think you said, DNA vaccines. Is that right? So can you just tell us a little bit about that journey from one system to another and how you chose where to go the way you've gone?
So all of these technologies, which we call platform technologies, they're very adaptable. You can use them to make lots of different vaccines. They all carry the genetic instructions to make the protein that we want the immune response against. DNA vaccines are very simple. They're just little circles of DNA with the instructions to make that protein, and that's all. And when you inject them into somebody, some of that DNA will get inside their cells. It's not a very efficient process, though.
Yeah.
So you get a bit of protein made, and that means you get a bit of an immune response. It isn't really very strong, and it's not enough to be particularly useful. So we tend to use viruses instead, and viruses are very good at getting inside cells.
Yeah,
So that's what they do. They get inside cells, and normally when it's a live virus, a pathogenic virus, it will then take over that cell, turn it into a factory to make lots of copies of the virus. But we've modified the virus when we make it as a vaccine, so it can't make lots of copies of itself. All it can do.
That's a really important point, Sarah. We should perhaps emphasize that with the audience, because not everybody really gets that side of it, that you're not putting in a real full virus that's got the ability to go around infecting other people. It's a very, very disabled sort of system. Can you try and...
It's a bit like a DNA vaccine in a way, in that all it can do is get inside the cell and make the protein we've told it to make. But because it is a virus and the outside of the particle is a virus, it has the machinery that enables it to get into cells very efficiently, much more efficiently than a DNA vaccine does. But then once it's in there, all it can do is make the protein that we've told it to make. And so that's what we get the immune response against.
Right. It can't make new viruses to go and infect other cells. It's just, it's a one-shot sort of thing.
Yeah, yeah. And there are different types of viral vectors that we use. With the malaria work, we did a lot with the pox virus vector, so it's a relative, the smallpox vaccine. But then we started using adenoviruses. Problem with using a human adenovirus, though, as a vaccine is that if somebody's been infected with that particular type of adenovirus, they will have some antibodies against it already.
And they are quite common, aren't they, some of these viruses?
Some of them are, yes. So we call them different serotypes. There's quite a large number of them. In general, the smaller the number, the more common they are because they got discovered first. So adenovirus 5, which is used as a vaccine vector, that was discovered early on because it's quite common. And a lot of people have got antibodies to adenovirus 5. So if we use that to make a vector for people, some people have antibodies against it already, and that stops it getting into cells as efficiently. So it's not quite so good. It's a great vaccine for livestock, actually, because they don't normally get infected with adenovirus 5. And so we can use it to immunize cattle or sheep, but not so great for humans. So we solved that problem by using an adenovirus that doesn't normally spread amongst humans. It's one that spreads amongst chimpanzees.
Right. Our closest relative, yeah.
So people aren't being infected with it. People don't have antibodies already. And then we avoid that problem of the so-called pre-existing immunity, which makes it means that the vaccine works well in everybody.
I'm just a thought has occurred to me, Sarah, that I've never thought about before. Where did you go and get this chimpanzee vaccine? Did you have to go to a zoo and find it from the chimps, or was it something that was already in a collection somewhere?
Well, we got it from somebody who'd had it in a collection for quite a number of years already, but it did come from a chimp that I think was in a zoo originally.
Right.
People in the past have gone around just virus hunting and collecting the viruses from the chimpanzees, but it came to us by somebody who already isolated it. And we don't actually use the whole adenovirus itself. We never use the pathogen. We don't use the adenovirus. We just use the DNA.
Yes, absolutely. Was it? I mean, it's casting your mind back. Was it quite a painful and long process to turn the chimpanzee virus into a vector, or was there a fairly straightforward pathway to get from?
Well, we knew what we needed to do to it. In terms of taking out the genes that may mean it can make more copies of itself when it's a pathogenic virus. So we had a template to follow because this had been done before, but the actual techniques for doing it at the time in the 90s and early 2000s were quite slow. These days we probably just order it as a synthetic piece of DNA, quite a big synthetic piece of DNA, but these days we can just send off an email and say, I want this synthesized, please, which would make life a lot easier. Then we were having to sort of painstakingly take out this bit and then change that bit. But we got it to the point where it was what we wanted and ready to add genes in. And then those genes will be whatever we want the immune response against. So a protein from the malaria parasite, a protein from the Nipah virus. And for some of these pathogens, it's a really big problem to know what to use.
Yeah.
Malaria has many genes, and it expresses different genes at different parts of the life cycle.
Whereas the viruses, like in fact these CoVs, it's relatively straightforward to know what you should probably put in, I guess.
Yeah. So with what we call the outbreak pathogens or the emerging pathogens, the viruses that occasionally cause outbreaks, and in this case a pandemic, they tend to be much simpler viruses. And there's usually not much of a problem to decide what the antigen is going to be. It's whatever's on the surface of the virus.
Yeah.
And coronaviruses only have one protein on the surface of the virus. So that decision was a very easy one to make. And obviously everybody's made the same decision last year. All the vaccine developers are using the spike protein because it's exactly the right thing to do. And it's that didn't take any time to decide on at all.
And in fact, you'd already been working on a reasonably close relative, the MERS coronavirus, hadn't you? Do you want to tell us a little bit of how you got into that and where you got to by the time the COVID-19 came?
Yeah, so I've been working on the early development of vaccines against a few different emerging pathogen viruses, and one of them was the MERS coronavirus. It's caused outbreaks in the Middle East, big outbreak in South Korea, in a number of hospitals a few years ago, but mainly cases in the Middle East. And it's a coronavirus, so we'd already got familiarity with making a vaccine against a coronavirus. Again, we use this gene for the spike protein, we put it into our adenovirus. We actually did some preclinical studies. We immunized monkeys and then we exposed them to the MERS coronavirus. We showed they were protected, they didn't become ill. We'd made it ready for clinical trials. So we're very lucky at the University of Oxford on this campus to have our own manufacturing facility.
Yeah.
It's MHRA approved and licensed. And
that's really unusual, I think, isn't it, for an academic lab to do that?
Very, very unusual. We go through the whole process of starting from very clean preparations of the DNA and going through the manufacturing, which means we can produce a vaccine that we can use in clinical trials. And we've done that with the MERS vaccine. And we conducted a first clinical trial in the UK, showing that we got antibody responses and T-cell responses against the spike protein. We'd gone on to do a second clinical trial in Saudi Arabia with collaborators there, which was leading towards a larger trial, which we would then hope to look at the rollout of that vaccine and testing efficacy of that vaccine. That's been on pause for a while because
I can imagine why, yes.
Yeah, all concentrating on the pandemic. But because we got this experience of using a coronavirus spike protein in a vaccine, showing it protected monkeys, showing it was safe in humans, we got good immune responses, the antibodies and the T cells, which is what we wanted to see. We knew immediately that a new coronavirus starts to cause disease, that we could do the same thing again.
Yeah, had it ever crossed your mind before COVID-19 came up that another SARS type thing might hit us, or were you just focusing on MERS and
well, it's always, I've always wondered if the original SARS would come back.
Yes, true.
Because it's probably there in bats. It may have gone through some mammalian species, started to infect people, caused an outbreak, which I'm sure people will be familiar with. A bit different from SARS-CoV-2.
Yes.
It didn't spread as rapidly. People had symptoms before they were able to transmit the virus, so that meant that it was easier to contain. And there wasn't time to develop a vaccine against the original SARS virus, and the outbreak was ended, but that doesn't mean that the virus has gone for good.
Oh, absolutely right. That's a very good warning. I mean, there are loads of coronaviruses out there in bat world, aren't there? And
there are lots of different ones.
Yeah, no, it's a wonderful world to explore. If any of our listeners have friends or relatives think you're going to do some research, it's a very good field for the long term, I think. So Sarah, I wonder if we could move on from that. You've painted a beautiful picture of how Oxford over a couple of decades has put itself in a position, you leading a big chunk of this, to be able to respond very precisely and powerfully to SARS-CoV-2 when it came along. Then the next bit is how we actually got a vaccine candidate out there into clinical use so very quickly. Because I think up until this pandemic, we'd got used to the fact that the process of developing a new vaccine from the concept stage to clinical approval would take many, many years, just like most medicines do. It takes a very long time. And with a pandemic like this one, as people can see, that clearly wasn't going to be any use at all. It would all bound to be over by the time a vaccine being developed. So how did how did you manage it? What innovative approaches did you take to speed up that process, to compress it into such an impressively short time without compromising public safety issues and so on?
Well, there are a couple of things that helped us with that, things that happened in the past that we learned from. So one of them was working on the development of an Ebola vaccine a few years back with a 2014 outbreak in West Africa. And the team at Oxford were asked to help with the first in human clinical trials of a vaccine, which the aim was then to get into West Africa. And then this is a vaccine that hadn't been tested in humans before, but in monkeys, it was completely protective against Ebola. And there was a need to get this vaccine into clinical trials and see if it worked while the outbreak was still happening. And so I was part of a team that did the early work on testing the Ebola vaccine. That went very, very quickly. Very good response from the regulatory and the ethical committees who prioritised the applications. We started our phase 1 trial very fast. We got our safety and immunogenicity data. We showed that it was well tolerated. We were getting good antibody responses to the Ebola glycoprotein, which is what was needed, and then passed that information on to a group in Mali in West Africa who were just on the edge of the outbreak zone, and they started testing it in healthcare workers in Mali. That went very well, very quickly. What had to happen then was to get it into a phase 3 trial, and that's where you immunize a lot of people, and you then see if the vaccine protects them.
Right. Okay.
But then there was a big delay. So our early part of the first in human trial gone very, very quickly. It needed to go into a phase 3 trial, and then nobody could decide on the design of that phase 3 trial. Should it be a placebo-controlled trial where some people...
variations, aren't there, to those sorts
There are.
Yeah.
And Ebola is a highly fatal disease, and there was an endless discussion about what was most ethical to do. And in the end, we'd finished our trials by November of 2014, and the phase 3 trial didn't really start until late April of 2015.
Oh, gosh, yeah.
So that left me with the very strong impression, I'm sure other people as well, that it's no good just thinking about the first part of the clinical development process. You have to think about where you're going to go to next.
Yes.
So in that instance, somebody else had prepared the vaccine. It was actually being manufactured in preparation for a clinical trial that had been planned on a rather slow timescale. So fortunately, that early part of the work had been done. We did our first in human trial very quickly. Mali did their second trial very quickly, and then everything just stopped. So actually what happened was that vaccine didn't ever go into a phase 3 trial because it took such a long time. The outbreak was being brought under control, which was excellent news. And one vaccine was tested, but not this one. So
always be in that sort of cycle, couldn't you, of never quite catching up with the outbreak? It must be very frustrating from your point of view. You've done everything necessary to develop something you know is probably going to work, but without the ability to get it out. So what lessons did you learn from this?
Well, so that was teaching us that as soon as we start to work on an outbreak pathogen vaccine, we've got to think about the whole pathway to getting vaccine efficacy, not just the first part of it. Because if you don't have plans for being able to do your phase 3 trial, which is what had happened in Ebola, then it's no good just having great data from a phase 1 trial. That doesn't mean you have a vaccine that you can use on a wide scale. So that got us thinking about, the whole pathway of clinical development. Don't just concentrate on the first part, you need to go the whole way. The other thing that helped was thinking about disease X. So we've been working for a while on making vaccines against a number of pathogens which were on various priority lists. The WHO had a priority list and the UK government had a priority list. We were kind of working our way through the list, but then the WHO introduced the concept of disease X, which is the disease that we haven't seen before, and we need to respond very quickly. And we started to think about how we would be able to respond quickly in terms of using the technology, in terms of getting into vaccine manufacturing very quickly, because we know that our normal route pre-2020 would have been to make the vaccine in the research labs, do some animal testing, looks good, raise some more money, let's see if we can get it manufactured for a clinical trial, and then we'll do the clinical trial. And then to start the manufacturing process, you have to go back to the beginning and start all over again with very clean preparations of DNA. So we didn't want to do that. We wanted to be able to go straight from the idea to get something into our manufacturing facility so that we weren't wasting time and we weren't repeating things. So we had been doing some thinking about how to do that. We hadn't had the opportunity really to develop procedures properly and to test everything out, but at least we'd been thinking about how we could do this. And so when we first heard about SARS-CoV-2 and that it looked like it was a novel coronavirus, and we didn't actually know at the beginning if it was going to spread and if we'd need a vaccine.
No.
But we did know that if we were going to need a vaccine, we needed to move really quickly and then go as fast as we could.
Yeah.
So we decided to start making the vaccine and to get it into the manufacturing facility at the earliest opportunity, to prepare for the phase 1 trial, to talk to other manufacturers and be preparing for the later stage trials. And all of this was working at risk throughout the year. And that means making preparations for the later stages before you know if the first stages have worked. Mm. And that's how we saved so much time, was by planning everything from the beginning and having everything in place to be able to move to the next stage at the earliest opportunity. Whereas in the past, in vaccine development, we would have done one bit, assessed it,
and then paused and thought about it. I love that. I particularly love the idea that by doing a bit of careful planning ahead of time, you can actually save yourself a lot of time down. Yeah. And sort of scenario planning. If it goes this way, then we do that. Yeah, very, very, very nice. Now, I guess the costs of doing some of these processes at risk must have been quite daunting. So how did you spend a lot of time raising the funds to do that? How did you go about doing that?
Yeah, in the first few months of the year, yes. Because every stage we needed to add a naught onto the amount of money it was going to cost us to get through the next stage. And as I said, we had some unexpected funding requirements that we'd never dreamed we would need to pay for. So the difficult thing, not only we were trying to make a vaccine against a pandemic virus really quickly, we were doing it in a pandemic. And we had to work with social distancing in our research labs.
Oh yeah, tell me, yes.
And in our vaccine clinic.
Yeah.
So suddenly we had to reinvent how we run vaccination clinics, because the old model would have been to invite people who were interested in taking part to come to the clinic and maybe have quite a large group of them in a room and talk to them, give them the information, give them the opportunity to ask questions before they then have their individual screening visits with one of the doctors on the trials team. We couldn't do that anymore. And one of the things that we did end up spending some donor funding on was actually 2 mobile clinics. So we had to expand the amount of space available for the vaccination clinics by bringing in the kind of buildings that are used at festivals. They come in on the back of a line and you unfold them and then you make divisions inside. And we've got 2 of those, which is where we were able to see our volunteers, but keep people separate and keep people appropriately distanced. And that was something that we hadn't ever really thought we would need to do. We had to have a lot of people running around between different buildings because people had to be separated out, but then we needed to get samples from one place to another. So we had a lot of medical students who weren't really able to go and finish their training because the hospitals were concentrating on treating the COVID patients. So they came and helped us and just kind of directed people around and moved samples around. At one point, we had to hire a plane to fly vaccine in from Italy.
Wow.
We'd had our vaccine, a batch of it manufactured in Italy. We needed to get it delivered to keep our trials going, and there were no flights because all the flights were stopped.
Golly.
So we chartered a plane, and it was just kind of problem solving the whole year. There was the expected problems to solve about how do you move from manufacturing, getting the applications into the regulators, massive dossier of information that has to go to them. And that was helped by the regulators being very flexible and taking it as soon as something was finished, they would take it and start reviewing it, rather than what they would normally do was we would complete the whole dossier and then submit it in one go at the end. And then they'd have
that's something I'd like to explore a little bit more, because I've heard this is like MHRA and people like that. Is that right?
Yes.
So they really they themselves didn't just sit back and do their usual sort of bureaucratic thing. They actively engaged with you to make sure that all of these stages happened in lockstep as quickly as possible. Can you tell us how you built up that relationship? These people you already knew, or did you have to reach out to them from scratch?
No, there were people we already knew there, and we'd been doing this kind of early phase clinical trial for a while with ChAdOx1 vectored vaccines. And that was incredibly helpful because we had a template for all the documents that we needed to submit to them. And it wasn't a MERS vaccine this time, it was a SARS-CoV-2 vaccine, but basically all the paperwork was the same. So we just had to produce the same set of paperwork for the new vaccine. So we knew what we had to produce and they knew what to expect from us. And that really helped because there weren't any surprises in trying to work with a new regulator, for example. And they did what they call the rolling review. So they don't just review the application to do the clinical trial, they review a lot of information about how the vaccine's made, and they review all the preclinical studies that have been done on the vaccine as well. So as soon as each piece of information was available and complete, it went off to the regulators and they reviewed that part and came back with any questions. And it meant that by the time we had our final clinical trial application ready to go to them, they'd already done most of the work. And so that, the final submission only took a few days to review.
In some ways, it's the most encouraging aspect of the whole thing, that sort of administerium hasn't held you back. They've been proactively sort of working with you, which is great. And I think it's really important to emphasize this for our audience, because there has been in the social media and stuff, various, ill-considered speculations that somehow, because the regulator did their job much faster than they normally would, they somehow didn't do it properly. Well, they did. They just did it in a continuous way all the way through. There was no step they omitted that they would normally do. I think that's
absolutely right. Yes, all the information was submitted. They scrutinized everything that they normally would, and they just prioritized these vaccine studies. Put a lot of people working onto it and were able to respond to us very quickly. But it was a continual ongoing dialogue, which is quite unusual with the regulator. Normally you have a formal interaction when you're ready to submit something, but this was a kind of an ongoing process to make sure that we'd answered all the questions that they needed answered at the earliest opportunity.
That's fantastic. Well, now we're well into the pandemic, and we hope that we may be seeing a change in the landscape now that we've got the Oxford, Pfizer and other vaccines well advanced in deployment, at least in higher income countries. We of course begin to face other issues. And I wonder if you could give us your take on some of these things. I know it's a very fast moving field. So, whatever you say now you might want to say something different in 6 months' time because of the way events change. But the first sort of area I think that's in most people's minds at the moment are these variants of concern, such as obviously what many people call the UK variant or the Kent variant, B117, and then the South African B1351 and the Brazilian, the P1, and further ones emerging. What's your reading of how the emergence of these is going to impact on let's just concentrate at the moment on the development of the ChAdOx, the Oxford AstraZeneca vaccine, but then more broadly.
Sure. So the big difference with the UK variant or the Kent variant is not really anything to do with immunity to the virus. It's its ability to transmit between people very, very efficiently. So if we remember that this was a virus that hadn't been transmitted between people until just over a year ago, it's not surprising that it's adapted and it's become better at doing it. And that's what this particular variant has done. So it's changed itself a little bit from the virus that came out of the bat, maybe through another mammal, and then started to infect humans. And whenever you have a characteristic like very good transmission, that's something that virus will take over. And that's exactly what we've seen, very rapidly spreading virus now. But what we do know
is it's twice as transmissible, or almost twice.
Yeah. Some debate over whether the disease that's caused is more serious. Probably not.
No, I think
probably about the same, but certainly very much more transmissible. So harder to stop it once you get it in a population. It's taking over from other variants. The good news about it is that the immune response induced by the original vaccines works very well against this variant.
Yeah.
We actually have data from the UK vaccine rollout as well as the later stage data from our phase 3 trials, which was collected while the UK variant was the dominant variant in the UK, and we get good efficacy against the UK variant. So don't have particularly to worry about modifying the vaccine to be able to cope with that one. The South African variant is different. Yes. This has changed so that the antibodies induced by infection with the original virus or vaccination with the original vaccines don't bind as well to the South African variant. And it's not completely black and white, it's not all or nothing, but there's a reduction in the ability of the vaccine to protect against the South African variant. And what we have seen in some animal studies that we did was that if we vaccinate with the original vaccine and then expose the animals either to the UK variant or the South African variant, if it's the UK variant, those animals are completely protected, their lungs are clear, and they don't have any virus replication in their nose and throat, right, and the upper respiratory tract. Different story with the South African variant. The lungs are still clear, but there is some virus replicating in the upper respiratory tract, so in the nose and throat of the animal.
Okay. Well, that's an important thought, though, isn't it, to separate those two bits of the virus's life, well, in the body life cycle. The lung stuff is where things go horribly wrong and people get really sick, and the upper respiratory tract is where the virus is replicating to transmit to other people and maybe give you the milder symptoms. So what you're saying is, even though there's a reduction in the protection against the South African variant from the Oxford AstraZeneca vaccine, the protection against the serious part, the descent into the lungs and the complication you get in the lungs, is still pretty much as good as it is against the prototype strain. Well, that's very encouraging, I think.
Yeah, so what's important to remember is that we might not be preventing all disease, but we are preventing severe disease, and the vaccines will still prevent severe disease even with the South African variant. What we had a clinical study in South Africa. It was just 2,000 people and they were quite young, and we had very low efficacy against mild disease in that study, which fits with what we saw in the animal models, because we know we're not protecting against the mild disease where it's just in the upper respiratory tract and people have a cold essentially, but people weren't getting seriously ill. In that particular small study in the control group, nobody got seriously ill either, so we couldn't say what the protection against serious illness was, but we would expect to get very good protection against a severe disease that puts people in hospital. And that means if you can protect against a severe disease, you're saving the strain on the healthcare systems. Okay. You're stopping people having to go to hospital with these really serious illness, becoming seriously ill. It does mean, of course, that there will still be some virus transmission.
Right. Yes, exactly. What I found striking in the piece of work that, as you know, we did in collaboration with our colleagues on vaccines is that if people have had a full course of vaccine, their immune responses, at least the humoral response, the antibody responses against the South African strain are way better than just the convalescent people who've just recovered from natural infection. That's surprising. Well, I mean, not surprising. It means the vaccines are really good. They're better than just recovering from infection.
Yes.
And I guess the same is true for your studies as well, is it, Sarah? Have you seen the same sort of thing?
Particularly when there's a long interval between the two doses, you get very high antibody responses, and that means that we are going to see at least partial protection against the different variants, even with the original vaccine. We are working on a new version of the vaccine with the South African sequence in it.
Yes.
We know in preclinical studies testing in animals, we get good responses against the South African variant, as you would expect. That will be going into clinical trials over the summer. It's not certain though that we need to switch.
No, but at least you'll be in the right place. So that if we do need to switch, you're prepared, just like before. You've thought ahead and you've got it all there ready to go if you need to.
Yeah, absolutely. So we're still, thinking ahead, being prepared. We don't want to wait until the autumn and then discover that, we really should have made a vaccine. Variant. We're doing it now. It might not be needed, but it's there if we need it.
Oh, that's fantastic. Now, the second sort of issue that's coming up increasingly, and I think this is possibly the first time this sort of thing has come up, because there's so many issues between different vaccine manufacturers, commercial interests in not mixing things. But there is now a serious trials going on, indeed, not just thoughts about, but actually trialing, mixing and matching different vaccines at the initial immunization and the boost stage and so on. You're obviously involved in some of these studies. What's your feeling about how those are and the logic behind them and how they might go?
Well, we're in a really unusual situation in that within a year, we've had quite a number of different vaccines developed against the same disease, same pathogen, using different technologies from different manufacturers. It's very unusual to suddenly have so many new vaccines all available at the same time. And all the manufacturers have had supply problems at different times. And so we can't guarantee that if somebody's had the first dose of vaccine A, that vaccine A will be available in the clinic when it's time for them to have their second dose. So the first thing that it's really important to know for public health purposes is it safe and is it effective to give a person a different vaccine at the second dose that they had at the first dose? So that just for practical purposes, if the first one isn't available, they can have whatever's in the clinic. Now, we would absolutely expect that that will be safe and that it will be a good thing to do, because with heterologous prime boost immunization, that means using one type of vaccine for the first vaccine, different type for the second vaccination, the same antigen in both cases, delivered in different ways. That's something that I've worked on for a long time, and my colleagues have as well. And you tend to get very strong immune responses that way.
And it's focused at the thing that's common between the two, which is what you want, isn't it?
Yes, yes. It tends to be very good. At the moment, nobody's really trying to optimize vaccination schedules by finding that the best one to give first and the best one to give second. It's much more about, if you have to give a different one, is this still going to work? And it's got more complicated now because we have more vaccines being licensed and coming into use in the UK in particular, so that, you know, the number of permutations are growing. But my expectation is that we would be able to use any of the vaccines following any of the other vaccines, particularly if we give them a 3-month interval between them. But it's something that has to be formally tested, and that's something that's going on at the moment.
I guess this is based on sort of a bit of prejudice, a bit of knowledge, I suppose. The sort of vaccines that you develop, the viral vectored vaccines and the RNA type vaccines, the Pfizer and the Moderna type vaccines, they would be good at both stages, and they'd be particularly good in that first stage because they're able to elicit T cell responses as well as antibody responses. I suppose, on the other hand, the protein subunit type vaccines or the inactivated virus vaccines are unlikely to be as good at the priming stage, aren't they? Because they're not so good at generating that memory that you were talking about earlier on.
Yes.
Is that the right way of thinking about it?
Well, that's the way I think most immunologists would think about it. Of course, it needs to be tested, but something that has been used in the past is adenovirus to prime and a protein to boost.
Sounds
so
good.
An adenovirus vaccine first and then protein tends to lead to very strong antibody responses. So that could be very good. So I think the prospects are good, really. We need to get the data to submit to the regulators so that they're confident to recommend these kind of mixing and matching. But I think I can't think of any of the combinations that really would be disastrous. I think they're all going to be useful, and some of them might actually turn out to be really very effective.
Excellent. So now I've had a little reminder from our organisers that we need to be getting on to answering people's questions, and so I've started to look at them, and we've had a huge number, and they're all very good. Luckily, we've actually touched on several of them already. One that we haven't touched on is why some people make a better response than others. So this has come from Fran Ashcroft, who I'm sure you know. So she's asking, why do some people fail to make COVID antibodies after their first vaccination? And does that have implications for their T cell response? Do we have an insight into that?
I don't really know why some people don't make antibodies after the first vaccination. We do know that they almost certainly will after the second. So some people's immune
Yes.
systems seem to take a bit more of a kick to get started, but then after the second vaccination, which is always recommended, then those antibodies kick in. But it doesn't mean that they wouldn't be protected to some extent after the first vaccination, because if they then encounter the pathogen, you know, the prime that they've
memory will.
yes, is ready to do something.
No, I think that's right. I think that's absolutely right. It's actually interesting to see how much variation there is in people to various immunogens, isn't it? And then Artemis Gores, I hope that I pronounced that right, asks, is there a fundamental feature that all coronaviruses share that would trigger a broad spectrum immune response to any variant? So in other words, is there like a universal human coronavirus vaccine that you could imagine developing?
Unfortunately, I don't think it's going to be that easy. If instead of the spike protein, we think about the nuclear protein, which is on the inside of the virus complex with the viral RNA, that is much more conserved between the different strains or different types of coronaviruses, not completely, but more so. But if you use nuclear protein as a vaccine antigen, you tend not to get very good protective immune responses. The reason we use the spike protein is it's the spike that sticks to cells when the virus gets into the body. And if we can block that, then the virus can't infect. And we're aiming to induce antibodies that block the ability of the virus to infect cells. And it's only by using the spike protein that we can do that. Exactly. If we use nuclear protein, the nuclear protein isn't on the outside of the virus. We could get a good T cell response against nuclear protein, which could then act once the virus is inside a cell and starting to make more copies of itself. But that's a slightly later stage. So it probably won't be quite such an effective vaccine. It could be useful, but not as highly effective as the spike binding vaccines.
We have actually touched on most of the things that people are asking. A couple of people are asking about the Moderna and the Pfizer vaccines, because this is a technology that actually caught me a bit by surprise. When I first heard that people were going to be doing these RNA things in scale and immunizing people, I thought that's just not practical. But actually, they've done an amazing job, haven't they? Do you want to, from your perspective as somebody who's not doing that type of vaccine, do you want any reflections on this? I think a lot of people think that somehow we don't like it when they're working, but it's not like that at all, is it?
No, we've said from the very beginning that we need as many vaccines against this disease as possible. It's great news whenever we get a new vaccine license. We need diversity in the vaccines that are being used because of the supply issues. And as I said, all the manufacturers have had a certain amount of supply problems. So if we put all our eggs in one basket with one technology, we could be really going down the wrong route. So RNA vaccines haven't been used very widely before this year. They've been in development for a while. The big problem is that RNA is naturally a very unstable molecule. It's something that's only supposed to be present in the body for a really short time. So our genes are made of DNA, and when the body wants to make a protein, it copies it into RNA, and then that's what gets made into the protein. But then the RNA is degraded. Because you don't want it to carry on, otherwise there'd be no control. So the challenge has been to get the RNA into a stable form that you can use as a vaccine and inject into the body and get it into cells. And so there's been quite a lot of work on the chemistry of the RNA, modifying it somewhat to make it more stable, which is what's been done with the RNA vaccines. And the other part of it is finding the right way of encapsulating the RNA in lipid droplets so that it can then be carried into the body, get inside cells and do the job that it needs to do.
Yeah. I think that things like coronaviruses have evolved millions of years to do.
Yeah. I think there's probably still a bit more work on that to do, because the RNA vaccines do need frozen storage because they're still not stable unless they're kept frozen until quite shortly before they're used. I'm sure that those, that's another technical challenge that will be solved in the future. But at the moment, it's making RNA vaccines pretty difficult to deploy in much of the world. Whereas with the adenovirus vector vaccines, we can store them at refrigerated temperatures. They're based on DNA, which is inherently more stable than RNA. DNA viruses are quite thermostable. And so that's a good point to start from if you want a vaccine to transport with refrigeration rather than having to keep it frozen, and it makes it much more deployable for large parts of the world.
That's excellent. There's a couple of sort of questions. I think we'll wrap with these two, given the time. Stepping back a little bit, looking at more broader implications. One from Ella Fung. How has your experience of scientific collaboration this year been? And has it been different from previous years? And what research trends do you hope will remain post-COVID?
It's been absolutely a fantastic experience of collaboration this year. We've had collaborators in the US at NIH very early on. Lots of vaccine manufacturers in the UK worked with us to help us get the vaccine manufactured for trials. Of course, AstraZeneca became our partners because as a university, we're never going to be able to manufacture and license and sell a vaccine. And it was really important to us to find partners that shared our aims to have a vaccine available for low and middle income countries and not just the high income countries. And that was part of the arrangement with AstraZeneca, that they would work with us on that. And what they've done is transferred technology to many other manufacturers around the world. So they have supply in lots of different parts of the world, which is turning out to be really important in being able to get vaccines to more people, which is what we wanted to be able to achieve. So there are lots of collaborations with other scientists, with people who work in regulatory affairs, because we have to work with regulators in different countries, and it's been a really good experience. And I hope that we do have more of that. I think at the beginning, some of the press were picturing it as a race between different vaccine developers, and we'd always said it wasn't that, because we need all of these vaccines. We're not competing against the other vaccine developers. We're competing to get a vaccine licensed to deal with this pandemic. I think it's taken a while for some parts of the press in particular to really understand that.
They don't like that story, yeah.
No, they want there to be winners and losers, but that's not how we've ever seen it. We've seen it as we need to get these vaccines out, and we've worked with other vaccine developers to, just try to make sure there are as many doses as possible.
I think we're going to have to get close to wrapping up, so there are lots of questions, some of which, like on the Indian variants and so on, I'd be happy to discuss. So that's the cooker hood going off in the background, sorry. But can I close with taking a future look, Sarah? So obviously we may, thanks to all the fine work you and our colleagues have been doing, we may be beginning to see the light at the end of this pandemic tunnel, although it feels like we might have got there by the skin of our teeth in many ways. But what lessons do you think we need to learn from this experience to make sure we're better prepared for the next pandemic? And do you think some of the developments that are being thought about in Oxford might be part of that forward thinking?
So in Oxford, we're hoping to have a centre for pandemic preparedness to build on what we've done. What we haven't touched on today is the work done on the drug testing, the recovery trial, which the results on dexamethasone, all of that, very important to do quickly and share the results widely. Lot of work to do in making vaccines deployable rapidly, in vaccine manufacturing, vaccine technology, all of that kind of work. We've done well this year, but we could have done better if we'd had better ability to manufacture more of the vaccine early on. There's still work to do in that area. So I think we've learned a lot. We've made some great collaborations. There's more to do. And we hope to establish a centre for pandemic preparedness, which will allow us to build on where we've got to and be better prepared for the future.
Well, I totally agree with you, Sarah. I think what Oxford and your group, amongst others, has done has been astounding, and it's shown how a bit of foresight built on top of decades of careful scientific preparation can make a real difference in real time, and how there's really no excuse without with the right infrastructure and funding for us not to be able to lead for the next one and going forward. So thank you very much for your contribution, Sarah. That's been a very insightful glimpse of your work and the context. And we will try and answer some of the questions that we didn't have time to answer in this chat by email later on. But in the meantime, Sarah, thank you for your contribution, and we'll hand back to our moderator.
Thank you.
My goodness, that was so wonderful. Thank you both. Thank you, Professor Sarah Gilbert and Professor William James. This was such an illuminating conversation. And for all the tutorials I've attended over the past year to learn about this and learn how there are so many cross-Oxford collaborations and within medical sciences. I've never heard such an intimate conversation before. And I think that was so uniquely Oxford, and what a blessing. And thank you so much for all your work from here in New York in the United States. Thank you. Thank you, Magdalen alumnus Jonathan Turner, for kicking us off and for the important contributions you made at the crucial early phase of funding development of Oxford's COVID vaccine. To everyone who's in attendance from around the world, we're glad you're here. You may learn more about Professor Gilbert's work and how you can support it by following the website links on the closing slide. Return to Meeting Minds tomorrow for the final programming of the 2021 edition. The North America office will present Oxford Chancellor Lord Patten of Barnes in conversation with author Michael Sandel, Balliol, 1975, speaking about his book, The Tyranny of Merit. Until then, goodbye.